This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 342, recorded on June 19th, 2015. Hey, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
How are you on this fine day?
Feeling great.
It's really nice out, isn't it?
It looks nice out, but actually it's hot and humid.
I love it. Hot and humid. 28 degrees Celsius, 59% humidity.
Yeah, you can feel that when you get outside. You can feel it.
But it's going to rain, right? Today, tomorrow, and Sunday.
That'll be 100% humidity.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
How are you?
Dandy.
90 degrees?
94.
Oof. Humidity?
We are experiencing summer.
Right. What's your humidity index?
94, that's 34 Celsius. And hang on, let me get it here, because this is important. Oops, it just went up, 95. Last I looked, it was a mere 39% humidity.
That's not bad.
That's not too bad. Dew point is 61.
that's not bad.
That's the telling thing. All right?
Yeah.
A couple of puffy clouds. Otherwise, it's summertime.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
Where is here?
Here is Western Massachusetts, near the city of Springfield, which was the first Springfield in the country, by the way.
Interesting.
Really?
How many of them are there?
A lot.
Is one in every state?
I don't know if there's one in every state, but the reason The Simpsons chose Springfield as the name of their town is that it's such an incredibly common town name.
Right. And it was named because there's a spring in the field where you live?
Something like that. Well, there's actually a river and a waterfall, and of course, because of the waterfall, they were able to build the mills, and that's what made the city of Springfield. Right. But it's about the same weather here. It's 83 Fahrenheit, 28 C, and humid and cloudy.
That's right.
It's got an annoying broken cloud layer around 2,000 feet.
We have beautiful cumulus here, right, Dickson?
Yep.
Is that what they are?
Our clouds are nicer
Very
down
nice.
here, Alan.
Oh, they're definitely cumulus. They're just a heck of a lot of them.
Oh, right.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 72 degrees Fahrenheit, 22 degrees Celsius, humidity 61%, partly cloudy. It's beautiful.
See, the front already passed you. That's over here.
Yeah, that's right.
So
We
I'm
did have
curious, Kathy,
a
do you have a dew point that you can report?
Dew point is, it's not scrolling up, 58 degrees.
Huh.
Okay. Interesting.
All right.
Well, that sounds pretty good.
That's the weather. We could stop it right there.
Yep.
Yep.
Yeah.
That's it.
But no one would listen, so it's because we're not local enough. So let's do some follow-up. We have an email from Jenny, who does our timestamps for us. Thank you, Jenny. So cool that you did Ben and Nick's measles paper. This is 2 episodes ago. Ben and I have talked about how interesting it would be to put the influenza glycoproteins into the measles genome and do the subsequent mutagenesis, but Dr. Palese said he didn't want to go to jail. Yeah, so that was something we suggested. I'd like to submit a listener pick for consideration. In this video, Bill Gates discusses why the government should be funding initiatives to prepare for pandemic viral outbreaks that spread more readily than Ebola. So she sends a link to a YouTube video. This is the earliest pick ever on TWiV.
Right at the top of the episode.
First 10 minutes, but it's okay. We can do these things.
Yeah, it's a good episode. I mean, it's a good video. He talks about what scares him or what keeps him up at night. And
Oh, really? He's going to get infected, huh?
no, he's reasonably worried about, a virus. So
Right.
He probably doesn't stay awake at night worrying about money.
No.
I don't. Yeah.
Not likely.
If he does, he's worried about too much.
I mean, he would get good medical care, too, obviously.
Yeah, but that doesn't save your life. Remember, Steve Jobs died. And,
No, I think he's worried from a global standpoint, not from a personal standpoint.
He has
malaria and that kind of thing.
Yeah.
Right.
Tuberculosis.
Bill Gates has a demonstrated global consciousness, and that's appreciated.
That's true.
Yes.
Yeah, I don't think you should worry about viruses, the kind that make you sick. You shouldn't worry about it.
I was sitting at the TED Talk that he was at, and he released a jar of mosquitoes into the audience.
Oh, that's right. I remember you saying
Famous, yeah.
Very famous.
That's right. So he's not afraid of anything.
I'm sure they were very carefully bred mosquitoes.
I do. Yeah, I totally agree.
From the best families.
Alan, can you take the next one?
Yes, Wink writes, I am a Twix groupie. I love and listen to every minute. I have one minor request, at least for TWiV. When you dive deeply into a complicated study, maybe you could give introductory and concluding sentences meant for the non-PhDs in the audience. Alan would be great at that. Wink, not a wonk, Weinberg. Sorry, I wasn't supposed to use the last name.
Okay, we've used his, because I believe he's an MD, so MDs and PhDs get their names read on TWiV because they're professionals and they can deal with the criticism.
We protect the people who are not setting themselves up for it.
I agree with Wink's take on that too.
So
I try to throw in a little synopses to the extent that I can, but I'll try it.
You do.
And we can all try and think about that too.
Yeah,
right.
Caleb is listening.
Yeah, you guys should. You guys should. I agree. Rich Condit, take the next one, please.
Robin writes, limits to variability for Dr. Condit to deal with some challenges. Compute the Earth's magnetic field values, and there's a link to a site from the National Oceanographic and Atmospheric Administration on ways to compute the magnetic field values. It's a little weird. Then there he, Robin,
I think. Yes.
Also includes pollen allergy forecast for Gainesville, Florida. And this is another site where you can look at your pollen forecast. Pretty weird. And then commenting on the measles episode, the number of variants is constrained by the structure of the variants and the structure of the domain in which they operate. For viruses, the domain in which they operate includes aspects of infectivity such as transmissibility, receptors, and host defenses. The measles virus in its configuration must effectively elude unvaccinated host defenses while maintaining infectivity and transmissibility. One or another would be compromised by variation, hence the configuration is so strongly conserved. A nice summary of that work. The structure of the variants can be constraining if too big an evolutionary jump to a new variant is needed. That's why unicorns and pigs with wings are so rare.
Because you can't get there from here. Yep.
Right.
No, you can't.
Kathy, can you take the next one, please?
Sure. Todd writes, howdy, TWiVers. Loved your discussion on episode 340 of the recent paper looking at constraints on measles virus mutation. It reminded me of a post I wrote back in February looking at an argument that cropped up from the anti-vaccination side of things. With the measles outbreak at Disneyland spreading quickly, they blamed first the vaccine because it didn't work, then shifted to blaming the vaccinated for spreading vaccine strain measles. Fortunately, genetic analysis had already been done and identified the genotype responsible for the outbreak. In my post, I tried to distill info on genotypes, serotypes, and epitopes into something a layperson could grasp. If you have time, give it a look and let me know what I messed up. I added a link to the measles paper you discussed. Best, Todd, and his site is Harpocrates Speaks. P.S. In Boston, it's currently 68 Fahrenheit, 20 degrees Celsius, with a chance of showers and forecast for continued slightly below average temperatures ahead. So I looked at this, and I'm not sure I can offer any criticisms, but I really liked his picture of bolts for showing the possible antigenic variation of surface proteins. It was fabulous.
Yeah, I thought it was, I cannot look at it from a layperson's perspective, but I thought it was well written. I didn't see any boo-boos.
It's a bucket of bolts, Rich.
Bucket of bolts.
Bolts.
That was a long time ago.
Is this the fellow who does the formaldehyde and all that aluminum stuff? Right? All right.
Right.
Cognitive dissonance in action.
Yes.
Nice.
Hold on.
Dickson Despommier.
Yes, here we go. Curtis writes, "Hello, TWiV team. Regarding the measles paper, were the resulting mutant proteins examined to ensure that the inserts were translated into amino acids?
Mutant
and
proteins?
integrated?" That's what he said. "It seems possible to me that the insertions may have caused a fold in the viral RNA structure that could have affected translation. A review of the resulting amino acid sequences would rule out any such reaction. I bring up this question because a couple of years of playing around with RNA secondary structure simulations has taught me that some sequences will tolerate very little variation before an unwanted fold forms or vice versa. As always, I appreciate the time and work you guys put into the show. Keep it up."
All right, so let me just make a general statement. You cannot talk about mutant proteins because mutations are by definition in nucleic acids.
That's
I know it's picky, but when you write a textbook, you have to be consistent.
Boy, you do.
Now,
Nothing personal, Kurt. He does this to everybody.
So how do you describe proteins that are
altered,
or
changed, substituted,
Yeah,
whatever you
okay.
want.
Yeah.
So he's concerned that the insertions of the sequence would mess up the RNA structure, and therefore you wouldn't get a protein, and that's why you don't get a lot of viable virus in all these insertions. Is that my reading?
Or at least in some of them, yeah, that's the idea.
Yeah, I mean, it's possible, right? I mean, and then you wouldn't really get an indication of what is architecturally constrained because you'd never make the protein, right?
Right.
But aren't we trying to make a case for measles as opposed to a lot of other viruses that have lots of variants, and they have the same problems here at this level that the others do? So
Yeah, you could argue that RNA, is
is that
going
a
to be
good argument? I think?
But I mean, I would guess that not every insertion is going to mess up the RNA in this way, right?
Yeah,
I would
mean,
guess-
what Kurt is asking for is, did they sequence the proteins
To make sure they were translated.
to make sure that they were actually?
Well, the
Big
problem
ones.
is that the non-viable viruses, you would have to in vitro translate the genome, right? And that's for all the insertions they made. That would be a huge amount of work.
Yeah, it's a good thing Kurt was not reviewer number 3 on this.
I'm not sure it would be worthwhile, right, to do that, just to show that the RNA structures were messed up or not.
I would certainly try to argue my way out of it first.
Yes.
You betcha.
But it's a good point that there could be
valid
other
point.
things. A valid point that there could be other things going on besides what you think.
Yeah.
But I think the overall paper, the overall concept of the paper still holds together because you'd have to, in order for that to be the explanation for all of the ones that didn't occur, you'd have to invoke some really weird RNA biology, I think.
Mm-hmm.
Yeah. So another general question that follows from this is that if you were to line up all of the viruses that you know of and put them on a scale of, no variants to thousands of variants, how would the world of virology look? I mean, would viruses fall into certain categories?
Yeah, well, I'd put HIV and influenza at the top there.
Absolutely.
Right?
Yeah,
It would look like a 5-dimensional plane on which
No, just a simple bar graph will do here. Thank you. Virus 1, how many variants? 1 through 5 or 20, and then, virus 2. And you keep going until you got them all down there, and then just line them all up according to the number of variants and see whether or not there might be some characteristics of their genomes that
see whether they cluster.
Exactly.
Yeah, I'd probably want to put it in at least 2 dimensions so you could cluster
clustering is what I'm
around
looking for.
viral type or
Sure.
species or
because
what have
these
you.
kinds of arguments are generic. I think you could make that criticism of any protein that's produced from any RNA molecule.
Dickson, I think this is a great idea, and I think you have yourself
Do it. Go,
a new
Just
project.
go at it.
Yes, go for
All
it.
you need is a
for it, yeah.
computer.
go for it.
You know what? That's exactly right. I mean, I think that data's out there. This is meta stuff. This
Those
is a
data.
master's thesis.
Yeah.
Yeah.
Maybe I can become a virologist after all.
Yeah,
Very
you can. We'll do your paper on TWiV.
Gee.
Yeah.
Dixon, hurry up.
Yes, I think I better hurry.
Dave writes, Dear TWiVniks, I'm sitting here listening to TWiV 340 and Rich's enthusiastic endorsement of Northern blots for RNA analysis, number 339. Bravo! But as I listened to TWiV 339 and then Rich's response, I found myself considering a combination of PCR-based quantification with an even more retro technique for RNA sizing by running sucrose gradients. That may take some explanation for at least some of your listeners. The RNA in each fraction could then be assayed by quantitative RT-PCR using primer sets targeted to any particular viral genome region. Having liquid samples that can be subdivided for multiple analyses often is easier than reprobing blots, and if you do it this way, you don't need radioisotopes. It's partly cloudy in 74. The dew point is 56, and we may get some more severe storms today in Hershey. Keep on TWiVing.
So I'm glad he did this because I had a Caleb moment after that discussion of northern blots, because there's probably a lot of listeners who don't know what northern blots are. So if I could follow up for Wink, was that his name?
Yeah, Wink.
Yes.
So let me see if I can do northern blot. You run a gel. You run your RNA on a gel.
What's a gel, Rich?
Yeah, what's a gel?
You electrophoresis it.
What's
You
an
elect-
electrophoresis?
You don't run it.
Electrophoresis. Yeah, okay.
Using
Okay.
RNase-free reagents, of course.
We should do a virology 101 sometime.
Oh my gosh.
So a gel, let's go ahead and do this. A gel is made out of agarose, the same stuff that makes Jell-O, right?
No.
No.
And no?
No.
It's gelatin.
Gelatin
No,
is a
that's
protein.
gelatin. Okay.
And
Yeah.
Agarose comes from seaweed, but it's
All right, you're right.
effectively the same kind of concept.
Same kind of thing.
Yeah,
All right.
it's an inert
And you
matrix.
you pour this slab of agarose that looks kind of like Jell-O, and you put your sample embedded in the slab in one end and run an electric current in it that forces all the RNA is negatively charged, so it wants to migrate towards the positive electrode, and the agarose has a sieving effect. So the small guys go faster than the large guys, and they get separated in this gel according to their size. And then you usually have a whole mixture of RNAs, and you want to find yours, and you do that by using hybridization. Okay, so you take a radioactive version, the complement of the RNA that you're looking for, and basically you have to, in order to do this, you have to transfer the RNA from the gel to another solid support called a nitrocellulose filter, if you like. But it's just effectively a piece of paper, and you've transferred the RNA from the gel, so the separation is still the same, onto this solid support, and you soak that in your radioactive probe. Your radioactive probe finds where your sequence is on the separated RNAs and hybridizes to it, and then you can put a piece of film over that, and you can see the image on the film. Okay, so out of this, you can see on the film how far your RNA has migrated down the original gel. And from that, with appropriate markers, you can tell how big it was. You can also tell whether it's really one band, it's homogeneous, or a big smear, heterogeneous, or whatever. By the way, it's called a northern because the technique was originally developed for doing the same thing with DNA. And that was done by a guy named Ed Southern. And those became known as southern blots. And then when Jim Allwine did it for RNA, being a clever dude, he decided to call them northern blots. So the big question is, do you capitalize northern?
Well, southern for sure, right?
Yes.
Southern for sure.
Right.
It doesn't explain
I
it
don't
all.
think so.
I always capitalize northern as well as western, which is doing not even really an analogous assay, but it's a gel blot involving antibodies.
So the reason I was so crazy about northern blots, people have forgotten about these now, because all of these, or they haven't forgotten about them, but they don't do them, because there's all these other techniques involving amplification of one sort or another to look for the presence or absence of sequence. But those techniques, by and large, don't give you any structural information. They tell you whether or not a certain sequence is there, and it's usually, you're looking for usually a pretty small, a portion of a larger sequence, and they don't give you any structural information the same way a northern blot does. Now what
David
is suggesting here is a combination of a sizing technique and a quantitative technique.
Let me describe sucrose gradients.
Go for it.
Could I do
Okay.
the Western?
So you have a centrifuge tube, and you can do your sucrose gradients a couple of different ways, but you basically form a gradient of sucrose either in the centrifugation step itself, or more commonly, you might start by putting 30% sucrose at the bottom and then 20% in the middle and 10% at the top or something like that. And then you put your sample on that tube and you centrifuge it. And again, your sample will move through this tube based on its size. And so, as David is suggesting, you could then collect samples, and that's often done by poking a hole in the bottom of the tube and collecting drops, either counting them or having a fraction collector do it for you. And then you have individual fractions representing different slices across the tube. And then you could take those fractions and do multiple things with them. I'm done.
So
many hours of my thesis work.
One of the problems is that sucrose gradients don't have the same sort of resolution as gels do.
Right.
Okay. But nevertheless, I like his idea
and also,
here.
If you forget about them in the centrifuge, then your stuff just pellets.
But I like his idea here because what he wants to do, if the picture that I get out of this is that, northerns are nice, but they're not very quantitative. All right. They give you structural information, but they aren't all that quantitative. The PCR is nice because it's quantitative, but it doesn't give you structural information, and he's trying to get both. Do the sizing on a sucrose gradient. That way it's easier to get the RNA back than it would be out of a gel, and do the quantitative stuff on the RNA that's sized on the sucrose gradients.
Right.
As you described sucrose gradients, Kathy, I remember one of my early experiences as a graduate student watching my mentor, Joan Stites, drip a sucrose gradient, which she did. This was a SW 50.1, right? So it's a 5 mil gradient. She did by putting a little piece of adhesive tape on the bottom of the tube. You could do this back in those days. The nitrocellulose tubes were fragile enough. The new tubes are tougher. She put a little piece of adhesive tape on the bottom of the tube and just stick a needle just barely into the tube, syringe needle, and drip it out of that. And she was collecting 3-drop fractions out of this, moving a rack of tubes under it, all by hand. And she was talking to me while she was doing it. And I said, how the heck can she talk to me while she's doing this? And I looked over, and I realized she was counting on her fingers. Her fingers on the desk going 1, 2, 3.
Did she have the other finger on top of the tube to control the flow?
No. This bit with a needle. You control it just by the gauge of the needle.
Someone did that. Someone used to do that.
I would poke the needle in just to get the drop started, and I would gauge the amount of distance that I poked the needle in so that I had a good drop
and
rate.
Remember, as the sucrose density, the percent sucrose changes, it's going to change the speed, right?
Yeah.
So you have to be aware.
So for the first 3.5 years of my graduate work, I did so many sucrose gradients and also cesium gradients. There were some days when I dripped 18 gradients.
Ooh.
Yeah. So
cesium gradients were nice, though, because if you forgot those in the centrifuge, I mean, not that I ever forgot anything in the centrifuge, but if you did, it didn't matter. They would just get, better resolution. Right. It's a buoyancy assay.
So sucrose gradients have had a renaissance lately because a lot of people are looking at polysomes in cells, the RNAs with multiple ribosomes on them. And a sucrose gradient is perfect for that. And then you can do a PCR, as David suggested, or people are even now doing deep RNA sequencing across these gradients to see, what kind of RNAs are on there. Very cool. So Kathy, the way we make sucrose gradients, depending on what you want the starting and the finishing percentage to be, you put like 30% sucrose in a tube, you freeze it, and then when you thaw it, you get a perfect gradient. It's really
now you come out with that technique.
So I was going to tell you the John Mudd way of doing it is you would put in your 2 sucroses, one on top of the other. Then you'd stick in a Pasteur pipette, swirl it around.
Swirl it, yeah.
And then just go with whatever you got.
Finicky 2-tube thing with an itty-bitty stir bar. And
I had one of those.
Oh, yeah.
I had
those make nice gradients.
We still have that, Alan.
That's a gradient maker.
But now you found a shortcut.
We did. That's what happens with experience, Alan. You find ways to do things.
I used to find all kinds of shortcuts. I wish I'd found that one.
It wouldn't have made any difference. Yeah.
The rap on sucrose gradients is that nothing you can do will make them any better, and nothing you can do will make them any worse.
So back to Northerns. I have a student who was trying to do her last experiment, and we haven't done Northerns in a very long time, and it got to the point where she had to do one. She's been working on it for 3 months, I would say.
We've been hearing about it for a while.
Boy, and she's trying to do non-radioactive hybridization, and she's just having... She walked in today. She threw down a gel. She said, "Nothing is working," and it used to, and then she left.
Welcome to the real world.
That's science.
And you want to be a PhD?
Soon, but whether scientist is another story.
Well.
Let's see here. We are up to Alan Dove.
So, by the way, that previous letter was from David Spector, who is a professor emeritus at Penn State, since we're doing last names on PhDs.
That's right. Alan, you can
All
take
right.
the next
Yes.
one.
So I'll take the one from Jonathan. Hello, TWiVers. Just heard the recent discussion on lab safety and reporting. I thought Alan was going to steer the discussion towards the NASA reporting system for aviation accidents. I thought that it would be a good model system, especially because reporting helps to reduce the chance of federal involvement. Just thought this might help further the conversation. All is well here in the Charm City at 72 Fahrenheit with a possible 92 Fahrenheit and some thunderstorms. Love the show and that you make it accessible to the non-technical type. So the Charm City, by the way, is Baltimore, for those who didn't know.
I didn't know that.
I
I
was going
didn't
to
know.
ask. Okay.
Good.
And yes, in fact, Jack, that occurred to me. It's Cheers, Jack, but it's Jonathan writes.
I must have messed it up.
Okay, well, this is from Jonathan or Jack. Jack,
It's probably Jack, because he does sign it.
Yeah,
Jack.
that occurred to me as I felt I was already holding forth long enough. But there is, in aviation, there is this, what I think is an absolutely brilliant bit of data collection combined with social engineering done by NASA. Number of years ago, they realized that a major problem in really any heavily regulated industry, but they were looking at aviation, is that it naturally breeds a code of silence, right? Because you've got all these regulations about safety, and it's important that people follow them. Inevitably, people will screw up, and when they do, their first reflex is going to be to not tell another living soul. So there would be these NTSB investigations of airplane crashes, and it would come out in the course of the investigation, actually, 4 other airline pilots had previously done the same thing and it had gotten away with it. Mm-hmm. And then they'd say, well, it would have been nice if we'd known that this was going on and people were getting into this habit. So NASA said, all right, we're going to set this up, and it's important that it's NASA doing it because the FAA is the regulatory authority with teeth, whereas NASA is more of a research agency. They set up this database, and any pilot or flight crew member or air traffic controller who commits or sees some kind of a deviation can call up the NASA ASRS and report the whole thing, spill the beans, forgive me, NASA, for I've sinned. And then NASA will call them back and say, okay, we got your report, we're giving it number such and such in the database, and they then anonymize that report and put it into their publicly accessible database. Now, of course, the question is, why on earth would anybody do this? Well, there's a carrot attached, which is if you are involved in some kind of an incident and you report it in a timely manner to ASRS, that number that they give you, this report number that's in the database, is a slightly limited get-out-of-jail-free card. So if the FAA is also investigating or subsequently investigating and they come across what you did and they finally come to knock on your door, they say, hey, we saw you, we think you committed this deviation, you can say, yes, you're absolutely right, I did, and I reported it to ASRS, and here's my number. And they'll say, oh, well, that's interesting. Thank you for filing the report. Have a nice day. Whereas if you hadn't filed it with ASRS, they would say, we'll take your license, thank you.
Do you ever get it back?
Well, depends on what you did. Now, the immunity this grants is not unlimited. I mean, if you crash an airliner and kill 300 people, it doesn't matter if you file a NASA report. There are going to be some consequences. But this is this really collects an awful lot of reports about the kinds of things that often become trends that lead to accidents eventually. And they put out a newsletter that you can subscribe to for free that they'll do different topics. So it's been an incredibly useful system, and I think any industry, I think this would be great to have for BSL-3, BSL-4 safety, where people could report stuff that's, nobody got sick, nobody got caught, but this happened, and then you'd be able to spot these trends when they're still subclinical, so to speak.
I know, it's a good idea. I think that would help allay some of the fears that are out there.
Yeah.
You'd need an agency to do this.
Mm-hmm.
Right?
You would need an agency that's not the regulator responsible for it. And when I was thinking about this, I thought, well, NIH is kind of both.
I wonder if the National Academy could do this.
The National Academy would probably be the place to do that, yeah.
If we get to it, we've got stuff on the National Academy later.
Okay.
All right, Rich, could you take the next one? It's from Stephen.
Stephen writes, Dear TWiVsters, I have to disagree with Alan about the USA Today article. This was the article about, don't tell me. No, tell me.
Biolab safety.
Biolab safety, that's right. If this were an isolated article, I might agree that this is good reporting with a sensationalist spin added after the fact. But the reporters at USA Today knowingly went looking for news that they could put out as sensationalist yellow journalism. How do I know this? Not just because USA Today's headlines often sound like clickbait. This is the latest in a series of sensationalist articles on biosafety, mostly written by Allison Young. They have a page for the stories released over the last year, and he gives a link. Now, most of those are sub-articles from the article discussed last week, but plenty are earlier, and USA Today's sensationalist coverage goes back even before that. Many of those articles are built around finding incidents and exaggerating or completely making up the danger that they pose to the general public. So I have no doubt that the editor and journalists involved, if they know anything about what they're reporting on, went looking for an excuse for fear-mongering. Frankly, I have an image of USA Today's newsroom as run by a real-world J. Jonah Jameson. For those who don't know, that's the fictional newspaper editor who is constantly looking for some scrap of evidence that would cast Spider-Man in a bad light. Instead of turning Spider-Man from hero to villain, USA Today is villainizing scientists trying to limit disease. And since the staff at USA Today isn't fictional, they don't even get to be characters we love to hate. And we can't excuse USA Today for not knowing better, not being science specialists enough, or not having enough voices speaking out from the other side. Other media companies can take a measured look at biosafety incidents and don't try to pretend that scientists are about to kill every last one of us.
Okay.
So,
All right,
yeah.
Fair enough.
Yeah,
I
I mean,
like
if this is part of a pattern, I was evaluating the article by itself, but
Yeah, I didn't know there was a collection of all these things. Let's see. Yeah. What else have they done? Buffering.
I remember a USA Today headline from some time ago. It was during some scare that we were talking about. I forget when it was, but it was out of hand.
Here we go. Some of these articles. Deadly diseases lurking at Arizona biolabs. Tennessee houses exotic deadly viruses.
yeah, the headlines are certainly sensationalist, and I do actually recall.
Wow, there are a lot of them.
Yeah, I do actually recall a few of these earlier articles that were a bit out of bounds.
All right, thank you, Stephen. And the last one for Dickson.
Jim writes, I was especially impressed with Alan's comparison of ethyl and methyl alcohols to methyl and ethyl mercury in the group's short discussion about thimerosal. I was also not aware that the reason thimerosal is still used is to prevent contents contamination in multiple-use containers of vaccine. All this is easily understood by us lay folks and easy to pass on. Thanks for those 2 insights. Jim.
We haven't heard from Jim for a while.
Yeah.
Well,
The ethyl methyl alcohol thing was not. I had actually seen that analogy someplace else, so I borrowed that one.
It's okay. Pass it on.
But it's a good one, I think.
All right. Now we have a couple of snippets. These are some things that I've actually written about in the past few weeks that I thought our listeners would enjoy, because I know the audiences are different for readers and listeners. So there are 2. One is a paper published in Science towards the end of May. It's called A Virus That Infects a hyperthermophile encapsulates A-form DNA.
I hadn't heard about aDNA since college biochemistry.
There you go.
So this is all about viruses that infect hosts that live in extreme environments, and this particular one is a virus called Sulfolbolus islandicus, rod-shaped virus 2. It infects a hyperthermophilic acidophile. That means it grows at above 80 degrees and around 80 degrees at pH 3.
It's 80 degrees C.
Centigrade, yes, Celsius.
pH 3. Hot acid.
Hot acid. So this is an extremophile, and so the microbe lives there, right? This is an archaean. And so the question here is, how do these viruses survive the heat and the acid? So so the assumption is that we should look at the structure of these viruses, and we'll learn something. So they took this virus and determined the structure of these particles, which are rod-shaped. This is a DNA-containing virus by cryo-electron microscopy and image reconstruction. And they have wonderful images of this lovely rod-shaped virus, which, looks to an approximation like tobacco mosaic virus, which is an RNA virus with a helical nucleocapsid. But here we have the DNA, which is wrapped in a helical manner by this protein. And there are two interesting things that pop out of this, which I thought were cool. One is that this protein, the capsid of the virus, so basically the virus consists of the DNA in this protein coding it. The capsid is made up of a single protein, repeated many times. In solution, in the absence of DNA, over half of this protein is disordered, so you can't see any structure when you go to study it. But when it's bound to DNA, that disordered part suddenly becomes ordered into alpha helices that wrap around the DNA.
So it sticks when in the presence of DNA, it sticks itself together like that toy you have in your office
That's right.
coils up with the magnets.
That's right. And the second thing is that this DNA when it's bound to this protein is in the A form, which is very unusual. Most of us have B form DNA in us.
Right.
And these are double-stranded DNAs. They look similar, but they have differences in the dimensions of the helices and the turns and the grooves and so forth.
I think of A-form DNA as if you take B-form DNA and kind of slide the two halves of the helix relative to each other.
Yeah,
so it changes the pitch a little bit, changes the size of the grooves and other stuff. But it's a relative. I don't know whether to call it a subtle difference or not, but it's still a double helix.
Yes,
put it that way.
Helix.
With slightly different dimensions.
It's kind of compressed.
Mm-hmm. Yeah, right? I was going to say just kind of squashing it.
Yeah, it's like if you take a DNA double helix where the bases are paired perpendicular, so you're holding it upright in your classic Watson and Crick pose, looking dramatic, and the base pairs are horizontal in front of you, and if you just crush the whole thing down and force the base pairs to kind of tilt and angle,
right.
Then you'd be getting toward aDNA.
Now, why, you may ask, is any of this have to do with living in acid at high temperatures? Well, we don't know the answer.
If you drop enough acid, things get distorted.
So there happens to be similar situation in gram-positive bacteria that form spores. So in some of these gram-positives, when conditions aren't good, low nutrients or whatever, they form spores that enable them to withstand extremes in the environment until things get better. And these gram-positive bacteria have small proteins that are unstructured in solution, but also become helical when they bind DNA. Okay, and the DNA converts to the A form when this happens. So this is to make a spore, a highly resistant spore. So it's functionally very similar. There's no similarity to the protein of this virus and to these proteins in these spore formers. But again, the idea is that in this virus, this highly disordered protein binds DNA, it becomes disordered, and they think that it may convert the DNA from bDNA to aDNA, like in the spore, which may be inherently more stable
Now, because
these
there's no
extremes.
similarity between the protein sequences, the supposition here, I guess, is that this is an example of convergent evolution rather than a borrowed mechanism.
Parallel?
Right, rather than the virus having
exactly.
inherited this from a spore-forming bacteria.
Probably.
It's more likely that the virus has simply encountered a similar problem to the one encountered by sporulating bacteria and has solved it by a similar mechanism.
Yeah, I think that's fair.
One thing that I wondered about, because it wasn't in this paper, and I just now remembered to look it up, is the size of the genome of this virus. And for the SERV, are we looking at SERV1 or SERV2? SERV2, it's 36 kilobases.
Right.
And the protein itself is 15.8 kilodaltons, so relatively small protein.
Yep.
And then I was wondering how many capsid proteins per virion, and they say the protein superhelix measures 4.3 nanometers and comprises 16.5 turns of bDNA. That still doesn't tell me how many protein molecules, but
you could figure it out, I think.
Yeah,
right?
but it's a pretty cool way to think about making this thing survive in this really hot acid. But they also point out that because the virus has to survive in the pH 5.6 to 6.5 cytoplasmic pH neutral in the bacteria itself, or the archaea itself, this virus has to really be adaptable to a wide range of pHs.
Right.
Yeah, I don't know what happens to this structure, but presumably it's different, and it's in bDNA, right, at normal conditions. But I don't know.
Right. Well, once it's in the cytoplasm, then it needs to unpackage this material anyway.
Yeah, maybe the unpacking then converts it to bDNA in the cytoplasm.
Yeah, as I recall, there was is there a different state of hydration in A and B DNA? I seem to recall that in vitro, the interconversion of A to B has something to do with hydration. I don't know if I'm disremembering that or not.
It's possible. I don't know.
Could they actually see the DNA in their reconstructions, or do they have the protein and they've modeled in the DNA?
They've modeled it.
Okay.
Yeah. Absolutely.
Well, one thing I like about these extremophiles is that it gives me confidence that no matter how badly humans screw up the planet, DNA will continue to replicate long after we're gone.
Maybe not our DNA, but some DNA will be
Some
there.
DNA.
Yes.
All right, the other snippet has to do with Ebolavirus. And this is a paper that was published in Lancet in April of this year. What month are we in? Lancet Infectious Diseases. Are we in June?
Yes.
Okay, thank you. It's called Long-Term Sequelae After Ebolavirus Disease in Bundibugyo, Uganda, a retrospective cohort study. Now, right now, there are a lot of survivors of Ebola virus infection, over 10,000, and there's never been that many around. And so the question is, are they going to have any long-term issues? You know, we talked about a guy who developed uveitis, an eye infection, a long time after clearing Ebola virus infection. There are lots of reports in the literature, kind of anecdotal, of people having various sequelae long-term. So here they did a very careful study where they took people who survived this 2007 outbreak. So this is now 29 months after the outbreak. And they identified 49 survivors. And they, as controls, they got 157 seronegative contacts. So these are individuals who had contact with infected people but did not seroconvert, presumably didn't get infected. And they brought them in and they asked them questions about their health. Is there anything wrong? They took some blood for blood chemistries and so forth. And in the end, they find that these individuals have a substantially higher risk of developing various issues, including eye problems, pain in the eye, blurred vision, hearing loss, problems swallowing, problems sleeping, joint pains, muscle pains, neuropsychiatric issues, depression.
Huge diversity in these.
I know, it's like multiple organ systems.
It's like
right?
they're just generally, messed up or run down or something, and lots of different problems are
and a lot of these
surfacing.
people could not return to their jobs that they had,
Right.
Before being infected. So, and this is over 2 years, right? The virus. Now, they didn't look for virus in this study, and an interesting question is what's causing these symptoms? Is it a consequence of virus destroying the organs initially, or a combination of that in the immune response? Is there any virus anywhere, right? So we know in the eye and in the gonads the virus can persist. So these are interesting questions. But clearly these individuals, and there are lots of them now in West Africa, are going to be an additional burden on the health system. You're going to have to take care of them.
Yeah.
These are not trivial. I think they mentioned in one of these papers, these are not trivial, I don't feel well kinds of issues. These are serious, substantial, reportable health problems.
Well, and the issue of people not being able to return to work in particular, you're looking at countries where they can't afford to lose a lot of workers and still keep the economy going.
Right.
These are places that are where everybody's struggling to get by, and this is a big deal.
Yeah. So I think it's worth noting that, this is what we consider an acute virus infection, right? The virus comes, it relatively quickly replicates, and eventually the infection is cleared. But there are persistent consequences of this infection. It's not unique to Ebola. There are other viruses that do this as well, of course, but here we're just coming off of a big outbreak, so this is going to be an issue. And this is not the West Africa outbreak, of course. This is Uganda, but it's a different virus as well. But presumably there will be similar sequelae.
Right. And the commentary points out some of the shortcomings of this, not that this isn't good in itself, but that it is what it is. And it's data collected at one time point. They weren't able to include children. They did look for correlates of two severe markers of severe disease, and found that those seemed to correlate with people who had more of these sequelae. So we'll probably get a lot more of that kind of data out of this more recent outbreak.
I would think so, yeah.
And in related news, at least one of the nurses who was infected in Dallas has filed suit against the hospital for apparent sequelae of that illness, which, obviously when anybody files a suit, some people are going to say, oh, it's frivolous, and but I don't know. If she's certainly, if I were suffering any of these sequelae, I'd be pretty upset. Yeah. And I guess the question the courts would have to decide is whether the hospital was actually negligent or caused that, and that's an argument that I think is up in the air.
Yep. All right, one more thing before we get into our paper. I was alerted to, you guys all remember the ALS challenge, right?
Right.
You pour a bucket of ice water, and Rich Condit did it. He challenged me, I did it, and Alan Dove never responded to my challenge. But that's okay. Maybe he will respond to this one. This is a challenge similar to raise money for chronic fatigue syndrome. It's called the Chili ME Challenge. And on July 1st, Ian Lipkin and Mady Hornig, who are here, are going to do this challenge. And it involves... what's it involve? I've lost it here.
I looked at the site. I didn't immediately
Eating hot chili?
Eating hot chili, yes.
Okay.
You're supposed to take a video of yourself eating hot chili.
Lordy.
And then film your reaction to it. Yeah, I saw it somewhere, but it's not on this page.
This is like eating a hot chili pepper, not like a bowl of chili that you claim is hot.
Right.
I guess you could do it. You could pour chili sauce on your tongue or something, or drink it, or chug it. Whatever. Don't do things dangerous. This seems like a tough one. I don't mind pouring cold water on my head, but... And then they have a spiceometer here at the site, which shows how much money is donated, from bell pepper to poblano, jalapeno, Thai, and habanero. So anyway, there you go. Put the link in it, and you should consider participating. It's fun.
One thing I wanted to say with respect to this ME reminded me that one of the comments on your blog post already points out that many of the symptoms or things reported could also be related to, or the same kinds of things would be things that chronic fatigue syndrome patients or fibromyalgia patients would report. And so
Right.
Just
Yep.
I have a go back. We need a word for this. I looked up the relationship between hydration and A and B form DNA. And from a 2005 paper from Nina Pastor, I find in the introduction, the confirmation of DNA is sensitive to hydration, and gives a reference to Franklin, that's Rosalind Franklin, and Gosling, 1953. The fiber diffraction pattern of DNA changes with relative humidity at which the samples are prepared. Diffraction patterns were interpreted as the canonical A and B forms of DNA, the low and high water activity conformations, respectively.
Cool.
Dan.
Thank you, Rich.
There you go.
I would like to point out that 1953 was the year in which I was born. All right.
And you didn't remember that?
I did remember.
He had his own DNA problems to sort out.
What month was that paper published?
I just got rid of it. I don't know.
All right. We have a paper that came out a few weeks ago, and we had 3 requests to discuss it. One is from Ben, who is actually one of the authors on the paper. Love the podcast. Please keep them coming. Thought you might find our paper of interest. Ben is a PhD at Johns Hopkins. Justin also sent the link, and Marshall as well. And he says he's been listening to the show since episode 3. And he said, I recall hearing on this week's episode 339, Vincent and Alan joking about a device that would tell you what viruses you currently have in your body. And I came across this article, which is the one we're going to do. I'm no virologist. What is that, from Star Trek? I'm no virologist, Jim. And I don't understand everything in this structured abstract, but it seems like it could be used to do exactly what you joked about. Run the test every day, and whatever shows up that hasn't been seen before would be your viruses for the day. Is $25 a day worth it to know? So he wants to know what we think. So I thought we should do this. I wrote about this a few weeks ago, but let's do it. It's a really cool paper. It's called Comprehensive Serological Profiling of Human Populations Using a Synthetic Human Virome. And the first author is George Hsu. The last author is Stephen Elledge. And in there is, I have to say, an interesting author, Suzanne Vernon, who I know, who used to be at CDC and worked on ME/CFS, and since went to the ME/CSF initiative, where I suspect she was collecting some of the sera that were used in this study. Anyway, the idea is here to find out what virus infections you've had, and by looking at antibodies in your blood. And the way they do this...
Inherently, that's an old idea, but this is a new twist on it.
So usually you go to a doc and you say, I feel this, and the doc will say, okay, let's look for this virus in your blood. And you look for specific antibodies against a specific one. But here we want to do a massive screen of everything that you have to see what your history of infection has been. And the way they do it is really cool. They use bacteriophages, and they start by looking for all the viruses that are known to infect humans, and they look in a database. They find 206 species, over 1,000 different strains of viruses, and then they take the proteins encoded by these genomes, and they make oligonucleotide encoding 56 amino acid pieces with 28 residue overlap. So they'll go down a whole viral genome and make these 56 amino acid tiles with overlap. So they do this with each of these viruses, and then they clone the sequences into a bacteriophage. They use T7 bacteriophage. And then when the phage particles are made after putting this DNA into bacteria, the capsid will display these peptides on the surface. It's called phage display. And then you can take these phages, the whole library, you've got a library of all the human viral proteins on these phages. You mix them with a little bit of serum from people. And if there are antibodies in there that will bind these peptides, you can separate out the phages, and you can recover the antibodies by pulling on them. You can use beads to pull on the antibodies, and then you can ask what DNA is encoded in the phage, and then you know what viruses those antibodies are directed against.
So is this reverse immunology?
Hey! Reverse immunology. No, it's not.
Phage display, by the way, has been around. phage display has been around for quite a long time.
It has.
Yes.
It's just immunology, Alan.
Yes, I know.
All right, so they got, how many? 569 humans, donors, right? And
across 4 continents.
Right, and they put them through this this assay. And they detected antibodies to 62 of these 206 viruses. And 2 people had 84 different viruses. The average is, where's that graph?
Average is 10.
10, and the highest is like 25 or so, and the lowest looks like 1, right? Or maybe 0, I don't know. I don't believe anyone had 0, right? Maybe. Anyway, the winner, the highest percent or prevalence in these sera is human herpes virus 4, also known as Epstein-Barr virus. 88% prevalence, 33 different peptides. And that's followed quickly by rhinoviruses, adenoviruses, respiratory syncytial influenza, herpes virus 6, herpes 1, herpes 5, more influenzas, enteroviruses. Now we're in the 2-digit tens. Norwalk virus, 11%. And then in the single digits, parainfluenza, cowpox virus, hepatitis B, parvoviruses. The lowest of their series was a beta papillomavirus.
Are you in the supplemental data?
Yeah, I am. I went beyond that chart in the paper because I wanted to see, because they only have the top 20 or so, I think, there.
Do they? I haven't looked at the supplemental data. Do they report measles, mumps, rubella?
Let's see. Hang on there. Let's see. We have.
I had those same questions, Rich.
That's very interesting. Rubella's there, 3% though.
Yeah, right.
Seems low.
So this, yeah.
So the antibodies might not.
Measles is 1.7%.
Yeah.
What about pox?
They had cowpox in there, but they say in the text that they think that's probably a false positive.
Yeah.
Right. Because pox virus makes some thats that look like it's got some genes that mimic human proteins, in particular a complement binding protein.
I wondered if they had a population old enough to have had the smallpox vaccine, if they could pick that up. That's what I was wondering about.
Good point.
Yeah,
probably.
So they do stratify these data greater than 10 years and less than 10 years. And you can see just by the dots that young kids have fewer antibodies to different viruses, which is what you'd expect, right? As you get older, you get more infections. But they don't have pox. Yeah, that would be interesting to know. I mean, the other thing that's low is polio, which is, I think it was 30-something%. Mm-hmm. You know, everybody's immunized.
Yeah.
Right, that's why I was asking about measles, mumps, rubella.
Yeah, they're all low.
To me, those are controls.
Yeah,
right?
so in Table 1, they show the sensitivity and specificity numbers for a few samples of people who have known viral infections, HCV, HIV-1, etc.
It's pretty good, right?
They're pretty good, but the sensitivity is definitely a lot lower than the specificity, even on those cases where people have the virus. And I suspect there's just from the top line data that the average number of viruses was 10. I suspect there's a major sensitivity limit going on here, because I really don't believe that the average number of viruses people have had is 10.
No, I agree. I think at any one time you probably have 10 or 12 different viruses in you.
Right.
So you should have more. And a lot of these numbers are too low, right? Rotavirus is 1%. The noroviruses are...
Rhino?
11%. Rhinos were high. They were 70% plus. So, I mean, I think that this needs to be worked on in various ways, right? Because remember,
there's a couple of ways you can miss things. One is that the antibody response can wane over time. Mm-hmm. Okay, so you may have reasonable immunity, but just not high enough serum antibody concentrations, and it could be that technically they can tweak this thing to pick that up. But the other thing is that all of these you're looking at are linear epitopes. Okay, it's just 54 amino acids, and it doesn't pick up conformational epitopes where you're looking at two bits, two two peptides that are separated on the linear arrangement in a protein but come together in the three-dimensional conformation and make the antibody binding site, the epitope. So you would miss those.
These are also produced in bacteria, which are not going to glycosylate the proteins.
Right.
So
Right.
if you have a sugar moiety that is your main antigen, you're not going to pick that up.
Right. So none of this is to knock the paper. It's just saying
this is
there's stuff to work on.
They
Great piece of work.
They should
It's a
consider
start.
doing this library in an AAV vector, right? So get around that glycosylation issue.
Right.
But the conformational epitope's harder,
Right.
That's going to
Yes.
be a hard one to crack.
That's a hard one to crack.
Right. Steve Elledge replied on your blog to someone and said that combining Plato with this would get at most of the conformational antibodies, but not those that need PTMs. And I don't know what PTM stands for. But so all of these things are things that they're thinking about. They
Post-translational
discuss a lot of them.
modifications.
Yeah, okay. Yeah. One thing I wanted to point out was in this very early figure 1b, where they just list for you about 14 viruses and about, I don't know, maybe 40 individuals, some older than 10 years old and some less than 10 years old, that if you look at the adenovirus and also rhinovirus A, there's a lot more abundance of positive peptides than for the people older than 10 years. And so I think right there is some evidence of waning antibodies.
Yeah. Yep. So they also found that in general, each sample was restricted in what peptide it responded to. So 1 to 3 immunodominant peptides per sample, which is, I guess, surprising. They say it's unexpected that the antiviral B-cell response is so stereotyped across people. So, they have many, many different peptides from each virus in their library, but only a couple were recognized from each person.
I find that really interesting. I don't know quite what to make of it, but it's really interesting.
I guess for vaccine design, it could help, right? If you want
Sure.
to either make a new vaccine or improve one, you would focus on those. But again, they're the linear epitopes, right? There are also differences in these peptides recognized between populations. So some donors recognized one peptide and others recognized a different one. That's kind of interesting, right? I guess that you should expect that because the immune system has genetic variability. So, it's a matter of recombination and somatic mutation that generates the antibodies. So there's going to be population differences. And I also thought, what was the other thing that was cool? I've lost it now.
They identify a lot more antigenic peptides than what had
That's
previously been identified in the immune
That's right.
epitope database.
It's like 10 times more or something like that.
And likewise, or conversely, there are things that are in the immune epitope database that are not among the things that they identify.
Right.
Okay, so those could be conformational or
Right.
post-translational epitopes as well.
So they also point out you could do this with bacteria, you could do it with fungi, you could do it with protozoan
You could.
parasites, Dickson.
It wouldn't be wrong.
What do you think you would find? What would be the top hit, Dickson?
Well, it depends on where you got your serum from.
U.S.
Toxoplasma, by far.
Uganda,
Malaria.
Vietnam,
Malaria.
China,
Malaria. No, probably Toxoplasma.
Australia,
Toxo.
South America.
Now you've got Chagas to worry about, too. Malaria or Chagas.
Huh.
Yeah, those two things.
You should do it, Dickson. Write a grant.
He's already busy on another project. What was that?
Oh, sorry. I don't want to distract you.
I was going to line up all the viruses according to the number of variants.
Oh, that's right. That's right. Yeah, that's a great project.
Come
I like
on, we
that.
need to know.
So they say you can do this thing with a drop of blood, 50 microliters, and $25 per assay. So I would do this, but it's cool to know what's infected you. But is there some real use? And I think once it's honed and made better, it would be really interesting to do big population-based studies.
Well,
and
that's
see if,
the deal,
See if there's
yeah.
any correlations with some of these chronic diseases that are puzzling. And I think CFS is one of them. You look and you say, on a population basis,
Yeah.
is there any pattern,
sure.
right, in CFS patients compared to others that might explain
Yeah,
what combination of infections might trigger this?
Yeah, they're clearly heading in that direction. I mean, as we say, this is something that probably needs a little more work before you go setting it into the clinic.
Yeah.
Because the danger with something like this would be if people put too much stock in it and did one of these tests and it came back negative for a bunch of things you thought the patient might have, well, that doesn't necessarily mean it's really negative.
Yeah, exactly.
A positive would be a little more reliable, but I think if this continues to develop as I hope it will, we should reach a point within the next few years where it is possible to distribute this widely. And I think one of the things that we will see at that point is a whole lot of initial results that scare the bejesus out of everybody. Oh my God, people are running around with rabies or something like that. And it's going to turn out that there are viruses that we now are very afraid of that we will probably find out there are subclinical infections of, and maybe we should still be afraid of those viruses, but maybe there are also people who are surviving them without any complications, and we would want to study that.
By the way, they didn't pick up any rabies here.
They didn't pick up any rabies, which is actually a good sign.
How about West Nile?
Yeah, West Nile was at, it was on here. Where is it?
I'd like to get my profile done.
I do it, yeah.
I wonder what my pox profile looks like.
Right.
I think you have
That's
pox
just pretty-
antibodies.
I'll bet you. Oh yeah, I've been vaccinated like 7 times.
I would
And
expect my
swimming
polio
around
titers
with
to be
stuff.
higher than usual.
Well, I don't find West Nile. I thought I had seen it.
No,
Dickson.
no, this was a pre-recording conversation that we had.
Maybe
and
they didn't find West Nile.
and maybe they didn't, but they probably should have. Well,
Yeah.
with a low sample number and all over the world, perhaps
Right.
that's not likely.
The one virus that I was puzzled about, these little single-stranded DNA virus, the viruses, they're in everyone, according to serological surveys, and they don't pick it up here at all. They have
Dengue?
Targetanovirus, which is like
Yeah.
1% or so.
Right.
So, it has to be tweaked. A correction, they found twice as many epitopes as we thought. I had said tenfold, but it's twice, doubling the number of all previously identified epitopes.
Okay.
All right.
Right. And so they found, yeah.
Alan,
and-
summarize.
So this is a test that scans for every virus you might have ever had. It's still got some issues with it, but they've already discovered some interesting immunological stuff from it, including that people tend to have common responses to viruses, so they react to these public epitopes that are generating more of the response than other parts of the virus might. It's a very clever technique that, with a little bit more work, could become a major clinical tool.
I wanted to add 2 things. First of all, HIV positive samples had more infections than negatives,
Yes.
which you would expect, right? Because they're
And was reassuring to see.
And finally, donors outside the U.S. had higher frequencies of seropositivity. Higher CMV in Peru, Thailand, and South Africa, et cetera. So, they say we don't know why. It could be differences in densities, cultural practices, sanitation, genetic susceptibility. Interesting.
There's another use for this data, and that is that they could estimate herd immunity for various emerging infections, and then up or not up the vaccination rate if they had a vaccine for that particular agent, like, for instance, measles and polio and the like, where if there's a lot of people that are positive, then the herd immunity for that place is good, and then if it's low, you redouble your efforts for your vaccine programs.
Absolutely. One thing I would like to be done is to do this with viruses that are not known to infect people.
Yes.
Right, and just see, hey, look, at 10% of the population is positive for this
For
virus.
rinderpest.
Or whatever, right? Or insect viruses in wheat, et cetera. That would be cool.
Yeah.
Plant viruses?
Plant viruses. I bet this is very expensive to do, though, right?
Setting it up.
Yeah, making all those oligos, right?
Yeah.
They made 93,000, almost 94,200 MERS.
Amazing.
Yeah, I'm not sure how they calculated the $25 per test. Does that mean you eat the development costs, or you amortize them across how many thousands of tests?
I bet you eat the
I
development.
think you eat the development.
Because, standard, an oligo is about, how much is a 20 mer is about $4.
Right.
So this is 10 times that. So it's $40 times 94,000. They had to get a deal on this.
Well, plus, there's a lot of brain time that goes into deciding what you're going to make.
Doing the tests, too. I mean, that's a lot of labor.
One thing that this test isn't going to show you right now is the things for which we don't have sequence for. Things that the viruses that we don't know.
Right.
Mm-hmm.
Yeah, we won't know what we don't know.
Yeah,
right.
The unknown.
An unknown unknown.
Yeah, there you go.
But as this gets if this gets to the point of being in wide usage, it could, as Dickson pointed out, it could be very useful in tracking herd immunity, but also emerging infections.
Right.
Yeah.
You could detect them right as they emerge if you have wide enough distribution of this and develop a much faster response.
Sure.
Potentially.
Indeed.
I found it interesting that they got a whole page to write their abstract.
Yeah, they published a single page in the journal itself, and I took it home last night thinking that was going to be the whole article. I hadn't looked beyond the first page.
Oh, is that what they did? I downloaded it and I saw this research article summary. And it's one page, and it has a page number from Science, and then the research article doesn't have page numbers from Science, so it's only on. That's what the whole thing is supplementary data now.
Effectively.
Wow. Apparently.
Wow.
Yeah, I know.
Yeah, imagine, because then I had to do VPN and pull down the article.
So 1,000 years from now, when all that's left of our civilization is paper and people are digging this up and they pull this article, they're going to say, wow, they didn't expect much data for a science paper.
Right.
Just the one figure.
Just one figure. Yeah.
All right, let's do some email. First one is from Gian, who writes, Gian wrote at the beginning, an email response would suffice since I don't think my email would fit into the current topics of your program. However, I do think it fits very well into the topic, so we're going to read it. Hi, TWiV team. I'm Gian, a future MS biology student from the Philippines. I've been listening to your podcast for about 6 months now, and I've been inspired by much of what you discuss here. I recently finished my second degree, BS Biology, from university and am applying for grad school. I'm hoping to pursue virology, but resources and faculty from my university seem to be limiting. At times, I'm discouraged by some PhD students to pursue virology due to the aforementioned obstacles, but I don't see myself settling for anything less. I honestly do not know what to do to get into virology. I figured that making a thesis on virology would be a good start, but it seems difficult to even gather the materials for it. At times I hear about the works of undergrad students from developed countries, and it frustrates me that I cannot do anything remotely similar to what they do. Of course, I will not let these hinder my becoming a virologist. To cut things short, my questions are, what do I do to get into a great virology program? Are there means to make the program available to an average to above average but very driven students? What got me into virology was Professor Racaniello's YouTube series, which means I blame him for getting me into this limbo. Just kidding, but his videos and the podcast really did bring me into virology. It is now 36 Celsius here in Quezon City, Philippines, with an 80% chance of precipitation, winds of 6 kilometers per hour.
That's hot.
Yeah.
All right. So John wants to do is going to be a graduate student in the Philippines and has problems not getting a virology program, it seems, down there, right?
Yeah.
Right.
So what to do?
Well, I mean, the first thing that comes to my mind is you apply to virology graduate programs elsewhere in the world. They're certainly most programs in the United States take international students. And you should know, John, that when you enter a PhD program in the U.S., you get a tuition waiver and a stipend, so you don't have to pay for it. You actually make a wage that you can live on. So if you could get accepted to a program, then, once you get over the transportation hurdle, and I suppose, application fees to start with, you're in. But you got to have the academic, an appropriate academic record to start with. But
a good friend of mine in graduate school was from the Philippines.
There you go.
And I think he may be a TWiV listener. So, Ricky, if you're listening, write in and let us know what it takes to get a Philippine student to the U.S. Because obviously he ended up at Columbia and ended up doing quite well for himself, I think.
Well, what would happen if Gian got a PhD in some other science and then wanted to do a postdoc in virology somewhere else in the world? Would that work?
Sure. Lots of times now, I think people hiring postdocs are looking for some pretty specialized background. So that may be a little tougher now than it used to be, but it's not impossible.
On the other hand, you could think that they're looking for more cross-disciplinary background, and that it could be an advantage.
No.
And also, if you end up doing a program in the Philippines for a PhD, if you focus on something that will leave you with a marketable, useful skill for virology, the thing that pops to mind immediately is bioinformatics.
Absolutely.
If you've got a bioinformatics PhD and you want to be a virologist, you can definitely do that.
I think that's right.
In fact, if you've got a bioinformatics master's degree and you want to go into a virology PhD program, I think you'd be a shoo-in. But there are other types of skills like that I think could be very useful. If you look into the techniques that people are using and some of the techniques that we're talking about in papers we discuss, whether it's deep sequencing and managing those types of projects, if you can acquire skills that are going to be useful in a virology lab, then you are in much better shape to get into one.
What I like about this letter is the phrase, "I don't see myself settling for anything less."
Yes.
That, as we've heard many times before, is nowadays basically a prerequisite for success in this.
Yep.
Yeah.
Alan, could you take the next one, please?
Sure. Monica writes, Dear virologists, I came across the topic of using bacteriophages as an alternative to conventional antibacterial therapies as a tool in the fight against multi-resistant bacterial infections. I was wondering what you guys know and think about this approach. What would you foresee as the hurdles that need to be overcome to get this FDA approved? And is this a good idea in the first place? And of course, a big thank you for TWiV. It goes without saying, I'm a big fan. P.S. Here in Germany, the weather is currently rather wet and cold for this time of year. And you've talked about this on TWiV, right?
Yeah, with 2 different episodes. We'll put a link. We had one where the team just talked about it. Then I had a live episode, I think, in Manchester, UK, where I had some guests who actually do this. So you should check those out. We haven't actually talked about this here on TWiV at all.
I thought we had touched on it at some point.
Did we?
I think we have touched on it a couple. Yeah.
But it is being looked at in many places.
And has been looked at since the discovery of bacteriophage, since the early 20th century.
So here in the U.S., one topical application has been approved, a phage to destroy listeria in food preparation, surfaces in food preparation areas. So you spray it on and sort of sterilize the surface, at least with respect to those bacteria. It's probably first going to get topical use in human infections rather than injection type of applications for topical infections. But I think people are very excited about it. And approval by the FDA is tricky because part of the problem is you'll have to match the phage to your bacterial infection, and that's not the way drugs work. So you'd have to tailor the drug for each person in a way. So that's going to be a hurdle, and they're going to have to just find ways to get around that. But,
and delivery is a big problem.
Yeah.
Because phage is not going to survive in the bloodstream. It's going to get eaten.
It's very antigenic,
and it's not going to get through the, you can't give it orally, which is why I would agree topical applications would be first.
Rich Condit.
I just lost the document. Dang. Here, I'll get it back.
We're up to Anthony.
Yeah. Okay. Hang on.
Yeah, your icon comes and goes on the Google Doc today.
Is that right?
Yeah, I don't know what you have a wonky computer there.
Yeah, it's on its last legs. Okay, I'm almost there. Anthony writes, disease puts rare migratory parrot on the verge of extinction. This is a headline from the Australian, June 1st, 2015. Gives a link and says the last wild population of an Australian parrot on the brink of extinction has been hit with a potentially deadly disease leading to disastrous breeding season and fears the orange-bellied parrot's annual migration from Tasmania to Victoria and South Australia this winter could be its last. The Threatened Species Commissioner, Gregory Andrews, confirmed yesterday that he had been briefed on an outbreak of beak and feather disease in the only wild population of orange-bellied parrots already characterized as one of the world's rarest and most endangered species. Its wild population is believed to be as few as 50 birds, with a further 345 in a captive breeding program. And he's got a couple of other links here. Another link to an article in Avian Biotech, and it says, by the way, thanks for reading my letter on TWiV, on the TWiV of May 30th. When I wrote that wild birds were not found to be the cause of influenza in poultry, I meant in particular this current outbreak in the Midwest. Of course, migratory birds are in general the usual suspects. Thanks again for your wonderful shows with TWiV as the jewel of the crown. So this is a circovirus.
Yeah, look at that.
A small circular single-stranded DNA virus that is trashing these parrots. Too bad.
You know,
Probably want a vaccine.
Would be interesting to do the virus scan on animals too, right?
Yeah.
Just think about it. Collect some serum, and I'm sure there are huge collections everywhere. How about cows and chickens and pigs? You know, so, of course they don't live very long, so well, cows do, right? But not pigs and chickens.
They have the potential for transmitting things to us, though, because we eat them.
Kathy Spindler, can you take Robin's?
Sure. Robin writes, he sends a link, and that's the entire essence of the article. And it was 2 weeks ago that I clicked on this. Basically, it's a blog post by Mary Odom that are sort of generally anti-GMO. And her name caught my eye because one of the two Odom brothers was at the University of Georgia. And so, indeed, she's the daughter of one of those two, and she's in Gainesville, Florida.
Howard. She was Howard's daughter.
And so, yes, also known as Tom. And so, basically, it's a, as I say, kind of an anti-GMO argument, and I posted as a counter post something that I think we had on here before. It's American Radio Works Pros and Cons of GMOs, or at least when I saw it, maybe it wasn't from TWiV, but it was a quiz that you took, and you started out, if you were pro-GMO, then you read the anti for a particular aspect of it, and then you kept on going through, and at the end you tried to see whether it changed your opinion at all. And I have to say that when I read it, it didn't change my opinion at all. So I'm one of those people that, I already believe what I believe, and you're not going to change my mind.
Yeah, well, you're right.
Yes.
Yeah, that's right. Dickson, at the end of this article, they say, GMOs only move us further towards an unsustainable goal of continued growth for a global economy.
Dear. And the next email even speaks louder to that subject of ignorance, unfortunately. This is advocating a non-GMO table salt.
Go ahead, read the email, Dick.
Sure. Cindy writes, the hurdle to educating the public about science and genetically modified organisms is higher than you think. As a full-time mom, I'm baffled by the dramatic increase in food fear and pseudoscience among my cohort. The level of time, effort, and constant and near panic put towards GMOs, processing toxins, allergens, dyes, chemicals, etc. is very sad to observe. Anyway, I think my picture of the week speaks volumes to the depth of the lack of basic science knowledge out there. Non-GMO table salts.
I actually had a grinder of this on my table
Wow.
Do you really?
Yes. So
Great.
my wife went to a
sad.
yoga retreat up in the Berkshires for a little mini vacation for a couple of days, and she was hungry one night, and their kitchen was just closing. They gave her some food. She wanted to put some salt on it, but the kitchen had now closed, and so she went to the little the shop there, and the only salt they had was this stuff.
Not a GMO.
Same thing.
You should have
It
demanded
was
the
exactly
GMO.
the same Himalaya salt.
Himalaya.
And it's called the purest salt on
Himalaya.
earth. And the first thing you notice about it is that it's pink.
Right.
And so I'm looking at this, but she brought it home because, of course, it's this whole grinder of it, and she only needed a little bit of salt on her sandwich or whatever. And she said, "Well, here's this." And I looked at it, and purest salt on earth, it's pink.
Yeah.
How is it pure? Because sodium chloride is not pink. The claim, it turns out, is based on the fact that this is salt mined in the Himalayas, so it's dug out of the ground, meaning that it came from an ocean that was there 180 million years ago, or whenever.
There was nothing else in the ocean and nothing else in the dirt.
Therefore, this was before we started putting all these evil chemicals into the ocean, and so it must be the purest salt, even though it's got, the sulfur and everything from the mountainside. Not only is it non-GMO stamped on the front, it's also stamped kosher and gluten-free.
Nice.
Oh, for God's sake,
Yes.
gluten-free.
Nice.
Gluten-free
Well,
salt.
GMO, I mean, come on, you can't GMO salt,
Exactly,
right?
and you can't have it glutened either.
I guess it could be kosher, though, right?
Yeah, there is an issue with kosher salt, but
So,
No calories.
yeah.
so I didn't actually say this to Vincent when I first walked in, but I'm afraid I'm going to have to leave now because I have guests coming soon to my apartment.
Want to give us your pick?
My pick I sent to you. It's a series of
I'll get it here.
odd visuals. Google puts them up every now and then, but the reason why I put it there is not because of the oddness of all of that. I was attracted to one of the items, and that was a 3D printable cast that you could put over, let's say, a broken arm, which was absolutely tailor-made to that particular limb. And then with two electrodes, they would pass a current through it and as a result generate a magnetic field, which caused the bone to heal 40% faster.
Wow.
And I was impressed with that because I know that they've done some work here in orthopedics, putting metal coils around broken legs and things like this, and showing that bones actually do heal faster in the presence of a magnetic current. But in this case, now they're using the 3D printer to make a custom-made sleeve.
Found from somebody doing an autoplay video.
I just love it. I just love it.
There's no autoplay on this.
No, there's some other stuff on there though. There are 25 things that you didn't know that was my pick.
This
Okay,
looks
I'm
like-
getting an ad from Verizon coming.
No,
No,
every time you hit
This,
those damn things,
the link I put in should be good.
I think it is.
Do you have the link I stuck in the show notes?
Now the ad for Verizon has stopped. I have not clicked on a link. I have one window open right now.
Yeah, just
Yeah.
brainjet.com.
Yeah.
Did
Brainjet.
anybody else get
That's
the,
it, Brainjet.
No,
the ad?
I didn't get it.
I could hear it.
I
But I
didn't
didn't-
hear that yet, but I don't know where it was
Too
coming
bad.
from.
Too bad.
Okay.
All right, Dickson, thank you so much for
You're welcome.
participating. Have a good evening.
Sorry that I can't stay,
It's
but
okay. We'll see you next week.
my computer says it's down to 4% electricity, so that means I have to leave anyway.
Yeah. Yeah, you were going to go to sleep anyway, right?
No, but my computer would. So
It
anyway,
was from that. The autoplay video was from that brainjet.com.
Really? It's
I see where it is, yeah.
It didn't play on mine. Are you using Chrome or Safari or what?
I'm using Firefox.
Yeah,
I'm
that's
using
what I used
Chrome.
to.
Maybe I have a built-in autoplay thingy.
Anyway,
Thank you, Dickson.
Lady and gentlemen,
Bye.
goodbye.
All right. Take care of it.
Bye.
Yeah, see ya.
Okay. So getting back to Robin, did he send that because he is anti-GMO, or he just wanted to give us another view? Of course, we
I
don't
think
know.
Robin is just doing his usual sort of pull our chain thing. That's all.
Yeah. The
You know,
stream
Robin
of
is-
consciousness links.
Yes.
Yeah. Okay.
Right.
All right. Let's do some other picks of the week. Alan, what do you have?
I have a blog post by an editor I used to work with, Porva Mandaville. And this is about science journalism and the diversity problem in science journalism. So if you thought that there were diversity problems in science that needed to be addressed, well, it spreads into science journalism as well. And she's got a really good explanation of what the problem is and why it matters. And I just thought it was a well-written post.
It's great. It's really good. Yeah, I think the first couple paragraphs are great.
Yeah.
Yeah, it's very nice. Rich Condit, what do you have?
This is a Carl Zimmer article from the New York Times about a discovery of a new microbe that is, their thinking represents a, as it were, missing link between archaea and eukaryotes. Okay, so there's stuff in here that I wasn't aware of. I'm behind the times here in thinking that there are three essentially independent branches on the tree of life: bacteria, archaea, and eukaryotes. Mm-hmm. But apparently the revisionist perspective on this is that eukaryotes evolved from archaea, right?
Yep.
And so this is analysis of DNA from a sediment that is about 2 miles down in the Arctic ridge that's between Greenland and Scandinavia, near a thermal vent. And the DNA sequencing from this sediment reveals the presence of a novel organism that they are calling, what is it?
Lokiarchaeum.
Lokiarchaeum. And it looks like it is apparently an archaea, but it's got a lot of eukaryotic-like genes, in particular genes that could be for membrane remodeling and genes that could be for cytoskeleton, but clearly doesn't have genes that would indicate that it has a nucleus or mitochondria. So this is thought to perhaps represent, actually Carl Zimmer writes this very nicely, represent something that, as opposed to archaea and bacteria, could maybe sort of mush crawl around and gobble things up. And you can imagine ultimately gobbling up something that could become a mitochondria and forming a nucleus as a reaction to that, blah, blah, blah. At any rate, this is really pretty cool. It quotes Eugene Koonin in here as saying that this is a genuine breakthrough. And I trust Eugene, so there you go.
Yes, if he thinks it's a breakthrough, then it must be.
Then it is.
Yep.
Right.
Yeah, he's cool. Of course, we interviewed him a long time ago on TWiV.
Yep.
Still a great conversation.
Yeah.
Kathy, what do you have?
I picked two links that are about cat genetics. The first one is one that my brother sent to me because he said, now I understand calico cats. And so this is from a site called Veritasium, and it's a YouTube channel, and it has a really cool... partway down the page, you'll get to the YouTube video about calico cats, and the title of it is something about why women are stripy. But essentially, it explains X inactivation, the epigenetics of that, and so women would be stripy because if you could look at our skin in different cells at the 100-cell stage or so, one or the other X was inactivated in each cell, and then that got propagated throughout the body. And that same kind of phenomenon is responsible for calico cats. And then I remembered from teaching genetics that there's also an explanation of why Siamese cats have the coloration they do. And it's because they have a temperature-sensitive tyrosinase, and it doesn't function at the normal body temperature. And dark colors only appear where the animal is cooler, like the ears and the paws and basically the extremities. And the face is also cooler because of air passing through the sinuses, it says. Anyway, it's a different way of thinking about internet cat videos.
Yes.
And my former postdoc, Adriana, is an international cat show judge. And I said, I bet you get asked questions about these all the time. And she said, yeah. And she was really happy to get the Calico Cats video. It's just really got cool animation about transcription and exon activation and so forth. Yeah, so I highly recommend it.
It's cool. It's cool when you get explanations for these things, right? Mm-hmm. Yeah. Science behind what things look like. All right, I have two related picks all about podcasts. It's an article in Wired not too long ago. Podcasts are saving NPR. So it turns out that they're losing money, except that podcasts are putting them in the black. And that's cool because, as you know, here on TWiV, we like podcasts. Of course, NPRs are slightly different. They have a higher budget and a lot more people involved, which we have a couple people and no budget. We do pretty well. We're not saving anyone, I guess, but it's nice that this is getting some news. And then in the Times, slightly later, an article, Podcasting Blossoms, but in slow motion. And again, an article about the growth of podcasts. This one is focusing on a guy who used to do something called This American Life, and he started a podcast show. And his name is, I guess, Alex Blumberg is his name. Alex Blumberg, right? Yep. So he started a company called Gimlet Media, and he's doing podcasting. One of them I've been listening to. It's actually quite good. Let me see what the name of it is here on my phone. It is called Startup, where he basically walks around with a microphone and a recorder and throughout his day and records his efforts to raise money for his company. You know, he goes to pitch the thing to venture capitalists, and then he comes home and he talks to his wife, and I'm sure she really loves that he's walking in the door with a microphone and records what she says. But you know, if you've listened to any of these American Life type podcasts, Serial is another one that he's done. You know, he talks and then there's background. It's all edited extensively. It's very compelling because he does a good job. That's his style. And Serial is built in the same way. So he's raised $1.5 million. He's got 18 employees, which seems a bit much, but you know, if you appeal to a lot of people, you can raise serious money. And I think this shows that this is reaching the mainstream, right? Because these are all big organizations that are making podcasts. Personally, I think podcasting is all about the grassroots, like us, who are just doing it because we have a passion for something and people enjoy it. But hopefully we can ride on the coattails if this gets big. And I think the key is going to be getting in the dashboard of cars in an easy way. As you heard me say, Kathy, out in California, as soon as people can push a button and listen to a podcast without any work whatsoever, which is what you do for radio, then it will really, really become big. And hopefully TWiV will be along there with it as well. So we had a listener pick from Jennifer. We also have two here. One is from Neva, a possible pick for the Super TWiV Gang, the Kardashian Index, a measure of discrepant social media profile for scientists. This is an article in Genome Biology, and it's basically, in the era of social media, you know, there are different ways that a scientist can build their profile. It's not just publication anymore. You can promote yourself on social media. Danger here that this can give you too high a value, right? If you're big on social media, but you publish crappy papers, you know, kind of unbiased. So help quantify this. I propose the Kardashian Index, a measure of discrepancy between a scientist's social media profile and publication record based on the direct comparison of numbers of citations and Twitter followers.
Mm-hmm. So that would be the K Index. Right?
K Index, yeah. Numbers of citations so I'll have to calculate this because I just passed 8,000 Twitter followers.
Wow!
In Scotland, the Scottish folk pushed me over the top. Near lovely Buda, Texas, it's muggy, 70 degrees Fahrenheit, 10 days of scattered showers and thunder bumpers forecast. The cattle are lying down this morning.
And I had to look up what that meant, and supposedly cattle lie down when it's going to rain. And in fact, I found a link to an article in the Daily Mail that confirms that they really do predict rain.
They
or so the article says.
I thought they lie down a lot anyway, right?
Evidently, they lie down when it's cold also. That would make
warmer.
sense. Yeah.
All right. So Neva was giving us a little Texas stuff there, right? When you had to figure it out. I wouldn't have looked that up. I would just say, well, they're lying down. What does that mean? Okay. Thank you.
Foot and mouth disease.
Thank you, Neva. I will do it for TWiV 342. You can find this one, all the others, for free at TWiV.tv and also on iTunes and using your favorite podcatcher, which you can get for Android and iOS devices. Hopefully one day we will be on Spotify, because Spotify is now including podcasts in their offerings. And we're trying to get in there so you can listen to us alongside Arctic Monkeys. And, well, she's not there anymore. I forgot her name. She left Spotify. We'd love to get your questions and comments. Send them to TWiV at TWiV.tv. Dickson Despommier is gone, but he's here at Columbia University. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Do you still do sucrose gradients?
I have not
Really?
done them recently, no. We do cesium gradients for virus purification occasionally. I'm not sure I've even done any in Michigan, so it's been a while.
Yeah. Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
Have you done any sucrose gradients lately?
When was the last time I did a sucrose gradient? We use sucrose step gradients to purify virus.
Yeah.
And I don't know when the last one was, but it wasn't too long ago. It's within the last, 5 years, something like that. Yeah.
Alan Dove is at turbidplaque.com, also on Twitter. Thank you,
Always
Alan.
a pleasure.
And I'm not going to ask you.
The last sucrose gradient I did was probably in about 1996.
In my lab, right?
Yes.
I'm Vincent Racaniello. You can find me at virology.ws, and we
have you done any sucrose gradients lately?
Personally, no, but my student has done sucrose gradients in the last couple of months. Yeah,
and is trying to do northerns.
She's attempting to do northerns. Very frustrated, and that's science. There are frustrating times. If you can't deal with that, then you shouldn't be in science. You got to be able to deal with failure all the time. I just shrug and move on. No big deal. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.